So we're talking about purine synthesis, and there's de novo synthesis, in which the purines are produced from scratch, meaning there isn't any precursor purine unit. And purines are the nitrogenous bases. The purine rings are the nitrogenous bases with the two rings to it, and the purine nucleotides are the actual nucleotides with the phosphate and the sugar attached to it. And then we use the purine nucleosides, which just have the sugar attached to the base but none of the phosphate. And the nucleotides can be both deoxy, meaning DNA ones, and simple ribo, meaning they're the ones in RNA. And we'll be talking about all of this in the clarification needed. So when we're talking about the purine ring, it is simply the nitrogenous base, and we have two of those. It's the nitrogenous base, and it is adenine and it is guanine, and these are the bases. Without the rings attached, neither ribose, which is in the ribonucleotides, nor the deoxyribose, which is in the deoxyribonucleotides, and none of the phosphate either. There is no phosphate in the purine ring itself. So we're talking about de novo synthesis, and then we're talking about something known as the salvage pathway. And the salvage pathway is more about recycling what you have. Starting with the de novo synthesis, right? So in the de novo synthesis, the first step is important. Now the first step often comes up. We start with ribose 5-phosphate. So you have to remember that when we're talking about de novo synthesis of purine, we start with ribose 5-phosphate, which means a ribose is a 5 carbon sugar. 5-phosphate means that at the 5th position there is a phosphate group attached to it. So this is the initial substrate that we start with, and that is converted into PRPP. This means PR means 5-phosphoribosyl, which is telling you the same chemical structure as this initial part. So means this part is the same. Phosphoribosyl, it's the same thing. And this means pyrophosphate means 2 phosphates are attached somewhere, and it is actually 1 pyrophosphate. Means that the first carbon 2 phosphates are attached in series. And this first step is very important and quite often asked. And interesting, the enzyme is really easy. It is PRPP synthetase. Synthetase means synthesis requires ADP. There's also the use of a cofactor, which is magnesium. Other important stuff are the allosteric activators and the inhibitors, which is very important. The regulation is important of this synthesis. The inorganic phosphate is going to be the activator, whereas the end product of this pathway, which is either AMP, not adenine, AMP, which is adenosine monophosphate, and GMP, which is guanosine monophosphate, meaning the nucleotides, the ribonucleotides. They're going not the deoxy. I'll explain that later. And what they're going to be is that they're going to be the inhibitors, the allosteric inhibitors. Now back to what we were talking about. So the second step is PRPP being converted into phosphoribosylamine. So ribosyl, and then you write an amine to it, because there has been a transfer reaction, and this transfer reaction is known as PRPP glutamine. Amidotransferase. And the reason for this is that this enzyme will have two sites. One will bind to PRP, another will bind to glutamine. Glutamine is one of those amino acids that is 2 NH2s. And the amido group of the glutamine will be transferred from the glutamine to the PRPP. So the byproduct of this reaction would be that glutamine enters, and glutamate, which is 1 NH2 less than glutamine, is what leaves. And the important thing is that this is a regulatory step. So when they ask you for the regulation of purine synthesis, you mention this step, and you mention that this enzyme, which is PRPP glutamine amidotransferase, is under the allosteric regulation of AMP and GMP, which inhibit it. The thing not to be forgotten is that while these are the inhibitory influences, the PRPP, which is the substrate, is the opposite.